We're now at the first part of the protocol, sample preparation and fixation. Since the aim of MPX is to assess the cell surface proteome, we need to start by immobilizing the proteins through fixation. I want to highlight that any treatment that is done to your samples between the time of interest and fixation, such as dead cell removal, release treatments, or selection, could theoretically affect the spatial structure of the proteome. Keep that in mind when you're planning your experiment, and always try to limit the time until fixation. And of course, always treat samples that you want to compare with the same kind of sample preparation. On that same note, you can use either fresh cells or cryopreserved cells, as long as all of the samples that you want to compare have the same baseline. If you have questions regarding different cell preparations, you can always contact us at support. The user manual consists of several washing steps, and when you're washing live cells, it's important to be quite careful. The washing process involves addition of the wash buffer, pipetting up and down to mix, centrifuging your samples, and then removing the supernatant. When you centrifuge PCR tubes, you can use either PCR inserts for centrifuges or place the PCR tubes in a PCR rack and use microplate carrier adapters. After centrifugation has finished and it's time to aspirate the supernatant, it's very important to be careful when you do so and always following the liquid surface down as you're aspirating. Otherwise, you risk losing a lot of cells because the cell palette will not be visible through all parts of the MPX protocol. So now it's time to fix the cells. Fixation is carried out using PFA, and this is the step where you're immobilizing the cell surface proteome so that nothing changes throughout the protocol. And after fixation, you have the option to either store them in the fridge for short-term storage or freeze them down for a longer time. So now that the cells are fixed, you can start pipetting your samples more quickly, and I will show you more on how to do that in the following videos. So see you there.